Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled GLP-1 Agonist Development, Endpoint Strategies to Help Differentiate Your Drug. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who have registered on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions that generate high-quality clinical evidence for their pharmaceutical, biotech, and medical device partners. They offer comprehensive evidence generation solutions that combine eCOA, cardiac solutions, medical imaging, precision motion, and respiratory endpoints. Now I'd like to introduce our speakers for today's event. Dr. Jessica Emerson is a behavioral scientist with over 15 years of experience in behavioral and social science research, focusing on the role of technology in measuring and intervening on health behaviors to improve health among patients with obesity and diabetes. She also has extensive experience in eCOA database design and management. Dr. Mark W. Tangowski's current responsibilities include clinical development and trial experiences in SLD, MASH, and biliary cholangitis, Crohn's disease, perianal fistula and ulcerative colitis, osteoarthritis, rheumatoid arthritis, psoriatic arthritis and bone age, and women's health. And Judy Narisi is a seasoned healthcare professional with over 2 decades of experience in clinical research. Trained as a critical care RN, Judy's research journey began in 2004 as a study coordinator, progressing to safety event case management and ultimately event adjudication in 2006. She has held key roles at PharmaNet, Johnson & Johnson, and ICON. But now, without further ado, I'd like to hand the mic over to our first speaker for today, Dr. Jessica Emerson. You may begin when you're ready.
All right, I assume you can hear me, so I will get us started here.
Yes, thank you.
Good morning and afternoon, everyone, and thank you for joining us. As Ryan said, my name is Jessica Emerson, and I'm very glad to have the opportunity to speak to you today as part of this interesting and quite packed webinar about digital endpoint strategies for differentiating your drug in what is really quite a growing market of diabetes and obesity drug development. Before we get started, I want to give a bit of an intro to just set the stage here. So you're here because you're likely aware of the major public health threat that obesity represents worldwide. In the US alone, over 40% of us are obese. That's a body mass index above 30, and over 70% of us are overweight, and that's a BMI of over 25. This permeates a person's lived experience through societal pressures, stigma associated with weight, and of course, there's a broad range of medical complications that are really directly related to being overweight. So there's heart disease, diabetes, stroke risk, and many others. In response to this, we're seeing a rising interest in medications to support weight loss, including a family of GLP-1 receptor agonists that are already approved for weight loss, and they have a growing potential in other areas as well. So what's cool is we're very timely today in this discussion because the FDA just released a new draft guidance last month for drug development for weight reduction. And you'll hear from myself and each speaker on what the guidance recommends for industry in our respective areas of expertise. So over the next 45 minutes, we are going to take you through a range of considerations and digital endpoint solutions, highlighting the role that innovative technologies can play in helping to differentiate your drug. So if you'd like to move to the agenda slide. So on your screen here is our agenda. I'll start us off with my area of expertise in electronic clinical outcome assessments. So that's going to include patient-reported outcomes on handheld devices and other devices that rely on patient use, like glucose meters. I'll talk about some key considerations for trial designs to achieve differentiation in efficacy and safety endpoints, while also following agency guidance and ensuring trial success in terms of the participants' experience. Then we'll hear from Dr. Tengowski, who will walk us through some of the innovative imaging modalities that are applicable to differentiating how a weight loss drug works and how that drug is affecting lean muscle mass and body fat distribution. And then, of course, to close out, Judy Nerisi will help us in understanding the role for clinical endpoint adjudication in obesity drug development, and that includes an overview of adjudication of cardiovascular outcome assessments, and also the role for adjudication of adverse events of special interest, which are key components to a differentiation strategy. We'll try to leave some time for some Q&A as well. Okay, Ryan, you can take us to the title slide for my section now. Thank you. Okay, to start us off, let's talk ECOA, glucose monitoring, assessment selection, and key trial design features. Next slide. Okay, what is COA? Here we see 4 different acronyms. I'm going to start us out with some acronym slides so we can all get us on the same page. These are 4 components of COA. So COA stands for clinical outcome assessments, which are used to measure how a patient feels, functions, or survives. And they can be recorded by a patient or a clinician, a caregiver, or observer. And that's represented on the right in the first 3 examples. And then shown in number 4, these can also be performance related. Today, we're really going to focus mainly on number 1, which is the PRO, or patient-reported outcome assessment. And that, again, involves having patients or participants report their own responses. And then ECOA is simply the electronic version of this data collection. So, for example, electronic patient-reported outcomes, or ePro, that's when you have a participant answer survey questions on a tablet or handheld device. So for GLP-1 trials, electronic patient-reported outcomes are designed on smartphone apps to ask participants questions about their experience, their symptoms, their blood glucose meter readings, which we'll get into some more today. Okay, so on the next slide, I have a couple more acronyms for you. What is SMBG? Self-monitoring blood glucose, or SMBG, is the measurement of finger stick capillary blood glucose by a patient using a blood glucose meter. So the patient uses a lancet device to prick the skin, and then they apply a drop of blood to the glucose test strip, and that's inserted into the BGM device. Modern BGMs have memories to store the glucose values, and many also have features like Bluetooth connectivity, so you can sync data with smartphone apps, and that's, as suggested as in the image that you see here. So you can probably already imagine how these devices support quality data with Bluetooth transmitted readings. Just that simple automation has been shown to substantially increase compliance and accuracy relative to having participants, say, transcribe their readings onto paper. So in contrast to that moment in time reading, continuous glucose monitors capture real-time interstitial glucose every 1 to 15 minutes. The interstitial glucose value is mathematically converted to a corresponding capillary blood glucose value, and that just provides a more comprehensive assessment, really different assessment of glycemic control than BGM. And that's because the CGM data is more granular, it provides many more data points near continuously to show change over time. And so this allows researchers to evaluate key metrics, like we can look at the percent time in a target range, the time spent above or below a target range, glycemic variability. And these are all really important indicators of diabetes management and risk complications. On the next slide, as I mentioned earlier, we're very timely today with this webinar, as the FDA has just released a new draft guidance in January. So the guidance clarifies expectations for clinical trial endpoints. And before I focus on how COAs are called out in the guidance, I should note that, of course, it also touches on imaging and clinical adjudication. And so you'll hear from that about that in the next couple sections of this webinar as well. But what I'm excited about is that they are calling out a role for clinical outcome assessments in phase 3 trials. So recommendations in this guidance also include encouraging early FDA engagement to ensure alignment on the trial designs and COAs. You should consider adaptive trial designs and strong safety data strategies to streamline the process with regulatory agencies. And then lastly, somewhat of our focus today, right, is for competitive differentiation. You'll want to consider targeting specific patient subpopulations and emphasizing patient-centric benefits. So on the next slide, I'll go into a bit more on safety data and hypoglycemia reporting. So in a crowded GLP-1 market, safety data could actually be your key differentiator. So not only that, of course, though the regulatory agencies expect a robust safety strategy. So I just want to touch on some important features of one here. So the table on top presents 3 levels of hypoglycemia. Level 1 and 2 have associated blood glucose thresholds, and level 3 does not require a blood glucose reading to be defined. Actually, level 3 was previously called severe hypoglycemia. It's actually characterized by cognitive impairment such that it actually requires assistance for recovery. So in a clinical trial, case report forms for hypoglycemia reporting can be designed so that one CRF captures level 3 and another could capture documented hypoglycemia at levels 1 and 2. And if you consider those 2 forms to include events with BG readings and then the other one containing the severe events, then you logically have 4 possible reporting outcomes, right? So either neither of the 2 CRFs are relevant, maybe both of them are relevant, or just one or the other form. And that, of course, is based on the event in question. And so this is one way to meet minimum reporting requirements to satisfy regulatory safety reporting. So you can imagine this is also an opportunity for a robust patient-reported outcome framework to be considered with respect to the at-home reporting of the blood glucose readings and the presence of symptoms. Though in our experience, the regulatory authorities have not traditionally required the exact symptoms related to hypoglycemia events be included in these event reports, but just if the event was symptomatic or asymptomatic is what they tend to want to see. All of this data supports a comprehensive safety data collection strategy, and it's really important as well for clinical adjudication of level 3 severe hypoglycemia for both type 1 and type 2 diabetes programs. So on the next slide, I'd like to tell you about where the FDA thinking resides currently for how to use BGM and CGM in clinical trials. So the BGM, again, that's the finger prick, really represents a point-in-time assessment. And that's used in trials when symptoms of hypoglycemia occur to check whether thresholds are exceeded. And that's important, of course, for patient and participant treatment decisions, but then also the safety data itself. So in an integrated electronic setup where a BGM, say, transmits readings to a handheld device, and then that can automatically send data to the sites. Sites can then be notified when the event meets certain safety thresholds to collect any additional data needed for the safety reporting, and that's sort of what I just described in the previous slide. So note that when it comes to the FDA, well, the FDA accepts BGM data for documenting safety data on hypoglycemic events. Let's contrast that to the continuous glucose monitor data. That collects readings again every 1 to 15 minutes. And what CGM is really suited for is comparisons between arms of a study on aggregated metrics. For example, you can look at the continuous nature of the data. That allows for the time in range or comparison of trends over time. And I should note too, it's especially useful when it comes to nocturnal hypoglycemia, which can be asymptomatic. So then when it comes to the FDA guidance for using CGM, the consensus is really still evolving. So you'll want to have discussions with regulatory authorities. That's really appropriate when you are trying to define your CGM-based endpoints. What I want to really point out here is that there's no definitive approach for using CGM to replace BGM readings for safety reporting at this time. So on the next slide, I'd like to go beyond hypoglycemia in COAs. So there are hundreds of validated assessments that can be used to measure patient-reported and clinician-reported outcomes. Just a small sample of some common assessments are shown here in those top 2 boxes. For these to be valid, they have to be administered in a standardized way that's consistent with their original validation. So faithful migration from paper to electronic format requires several considerations, including how the text is shown on the screen, the instructions, the flow, and it really needs to be consistent with the validated survey itself. Now, when these assessments are collected electronically, we unlock some really important advantages. We're seeing higher quality data, improved accuracy and consistency in line with regulatory expectations. And some of those include contemporaneous and attributable data points. And that can be challenging without electronic features for at-home data collection when it comes to timestamps and other features of the alarms. On the bottom of this slide is just a sampling of potential targets to differentiate a GLP-1 drug. For example, just last month, a phase 2 trial was published in JAMA that showed significant impacts on alcohol consumption, for example. So I'll stay on this topic for the next slide as well. And you can go there. And so I'll zoom in on some common validated participant-reported outcome assessments with more potential targets for differentiation. What I'd like to point out here is the importance of investigating the subscales on the validated assessments. For example, the SF-36, there's assessments you can use for bodily pain. There are features of the 3-factor eating questionnaire, like cognitive restraint, that can prove to be key for eating-related or addictive behaviors. So, when looking to differentiate your drug based on a participant-reported outcome, having a validated scale is perhaps just an obvious first step for validity and for detecting an effect between treatment and placebo groups. What I'd like to really expand on in the next slide is the supporting infrastructure that makes it possible to detect the signal from the noise in subjective experiences, which is ultimately what a lot of PROs are designed to measure. Success here really requires an entire system to have the least room for error, minimize missing data, and activities that can fit seamlessly together in the overall trial experience. Considering the range of responsibilities that are shown here that ultimately fall on the participants, having a coordinated data collection method using integrated devices, that's one key foundation for overall trial success. That supports data quality and compliance and reduces participant burden, and that should also reduce attrition. And I'll get a bit more specific on the next slide too. So to give a bit more of a concrete example about what some of those seamless features should look like for successful differentiation when it comes to supporting the participant-reported outcomes, here are some practical design features that will reduce data noise and enhance data quality. First, on the left, we have the user experience from a survey design standpoint. That can literally have compliance built in by design in an electronic system. So you can use automated branching, reminders, appropriate timestamps, recall ranges. And in the middle, we have some examples of some engagement tools that will help participants know just right where they are in terms of fulfilling their reporting responsibilities. And then on the right, I can't stress this enough, 24/7 access to training content for participants is really key. You want to train them in order to reduce variability in question interpretation. The training should be, allows it to be standardized, and that standardizes how compliance is explained to participants. And think about it, that's really critical for a large global trial where even slight variations can just organically arise in the day-to-day of trial activities. So on the next slide, one last practical design consideration: when collecting at-home data is done electronically versus on paper, advanced analytics can flag early warning signs. So you can look at concerning submission trends by a particular participant or a set of participants at a site. Issues on the site side can be tracked for clinician-reported endpoints, or really just any other metric relevant to a given study design. These can be customized. I can't stress enough that the capabilities are there from a technology standpoint in terms of data oversight on advanced, well-designed dashboards. Plus, the regulatory guidance calls for a comprehensive data monitoring strategy. So that's going to bring me to just about the end of my section. So on the last slide, I want to wrap up some of the key considerations I've touched on today. They include a comprehensive and efficient ECOA strategy, and that blends efficacy and safety endpoint data into a seamless experience for participants and for sites. On the safety side, when efficacy may be similar, safety could be the key differentiator in some cases. So it's important to have someone like Dr. Strumpf, who has decades of experience in diabetes drug development, clinical practice in endocrinology, and that really translates to pragmatic input. Clario, Paul consults around these key pieces, from early protocol draft reviews to implementing CRF and adjudication strategies for hypoglycemia-related event reporting. Lastly, and importantly, the regulatory guidance really stresses training for participants, yes, and sites too. And of course, as I was just talking about, right, the nature of electronic data allows for customized and advanced analytics strategies for data monitoring. Okay, I think that concludes what I have for you, and I will now invite Dr. Tengowski to come on screen and talk to us about the different medical imaging modalities that can be leveraged in obesity development programs.
Thanks, Jessica. That was a really great talk. I think the importance of glucose for both efficacy and safety does translate into imaging when we look at some of the endpoints that imaging will tell us about where is weight loss occurring. So let's go on to my first slide, please. Thanks. So, you need to ask yourself, what kind of weight loss happens when you've got subjects that are on GLP-1 receptor agonists? And there's kind of two modes at the moment. I'm looking at obesity, where I'm wanting to shed unwanted fat, or I'm trying to modulate my glucose to bring my A1C level down. Both valid endpoints, both tested concepts, but we're going to really focus on the how imaging relates to the weight loss, because the data so far has suggested that you can see between 5 and 15 kilos over a year, and there is some differentiation of how much weight loss you expect. Now, you've got the weight scale, but you also have where is that weight loss occurring, and that's the question here about we're seeing weight loss that was attributed to lean muscle mass and not just fat. And really, we want to maximize that fat loss, minimizing the muscle loss. That should be an important goal in any of your investigations. We know that imaging can be used to look at the degree of fat and lean and those changes over time. We can also do imaging of large fat depots in the liver, in the abdominal cavity, around the heart, other organs as well. So don't ignore bulk fat as your endpoints. Skeletal health, I think, is really important in diabetics because of the possibility of they're older, they have maybe not great muscles to begin with, and the possibility of fracture risk is there. We want to monitor the bone performance. So the FDA draft guidance in designing an endpoint in a trial that uses imaging, we want that drug to be effective for weight reduction and maintenance. And after 1 year of treatment at that maintenance dose, that's where we're going to look at weight reduction. And that has to be between an investigational drug and a placebo-controlled group. And you need to have at least 5% difference for it to be significantly, statistically significant. And we know imaging methods, what their variability is, and then what's that sensitivity to change over these time periods. So let's go on and look at some more information about weight loss itself. So we mentioned earlier that weight loss includes both muscle and fat. And of course, we want the maximum amount of fat loss with the minimum amount of muscle loss. So what are we looking at here is the quantity of the changes in those pools, the composition of the muscle and the fat, health of the individual, have we given up any strength, and any of the functionality related to change over time. So we know that caloric restriction reduces protein synthesis, and that increases muscle proteolysis. Now, those changes are maladaptive, or what we would call bad changes. We want to reduce that. What makes comparing trials difficult is there's a lot of factors that go into this. But when we look at where are we seeing reductions in lean versus fat, if I'm looking at semaglutide, I can see between 40 and 60% total weight loss, and the tirzepatide is in the 15 to 25%. Now, that's an unfair comparison in this statement, but I want to lay out the groundwork that you need to do to say, what is my patient population that I plan to study? What are the specifics of the drug being tested? This is more mechanistic. What are the comorbidities of people coming in? Is this just BMI? Is it A1C? And then how can I measure lean muscle mass as a composite measure, knowing that a modality like DEXA also includes organs, bone, fluid, and the water in the fat tissue? And then is something like MRI maybe something I should be using to tease out those details? So our draft guidance is saying that in our pharmacologic trials, we should be shooting for 60 to 90% weight reduction, and we want that reduction to occur in the fat pool, not the lean mass, because too much lean mass could be considered adverse. So let's go on and talk about the specifics of imaging modalities here. There's quite a bit of methods. I'm going to mostly talk about DEXA, MRI, and I'm going to introduce CT, but not really go into a great deal on CT. We don't like to use CT because of all the radiation. But I do have different pools that I'm interested in. I can look at adipose tissue, and we used to think that adipose was, just fat without any real activity. It turns out that adipose itself is actually very biologically active, and there can be inflammatory mediators coming from different parts of the body where adipose is stored close to the organs. So there's a real interest in looking at adipose, and we can look at the direct drug action, whether I'm looking at total body fat or regional mass, or if I'm doing it with MRI or DEXA. And I can also look at the SAT versus VAT component, because we know there's a difference in adiposity in the abdominal region, that's important biologically. From a functional perspective, muscle, I can use, again, I can look at DEXA, I can use MRI doing either total body or regional analysis. I tend to look at the abdomen, mostly so I can look at the spinal muscles, or I can look at the thigh. If I needed to look at the deltoid muscle, I can use that as well, but it's mostly the thigh, and the gastrocnemius is my muscles of interest when I'm looking at this kind of an indirect drug action, or the changes related to mechanical unloading. Tissue fat content, here we're thinking about, we've done many studies that's standardized to do liver fat, but I can also look at intramuscular fat and the changes in the sub-Q to the fat within the muscle or the fat between the muscles if I use a detailed MRI analysis. Because we have body composition changes and the theory that overweight people have a lot of muscle, and to say that they have they're big boned, that's actually kind of true. There's the mechanical loading of the skeleton changes when I lose weight. Hopefully I'm losing fat and not muscle, but I can look at my changes in bone mineral density over time and determine if I'm getting into a kind of a safety observation with too much bone loss, because if I'm looking at older people, frailty, fracture risk, all those things factor in. I can also look at the bone in very great detail by using quantitative CT, and you can do that in the spine, you can do that in the femur and the hip. Those are all great endpoints for you to look at. What are you seeing with the body weight reduction? And then not only are you seeing a reduction in fat content, but what percentage of it was fat and what percentage of it was muscle. So let's go on and think about, the weight loss that occurs here. We want weight loss to be adaptive. And this is not a foreign thought at the moment, because we know that complementary imaging like MRI has shown reductions in muscle volume, and over time that can be expected because of aging, disease status, or the weight loss achieved. I mean, some of these agonists are, producing, 40, 30, 40 pounds of loss, and of course, some of that's going to be muscle. We want to minimize that as much as we can. When we do have weight loss, we also hope to have increased insulin sensitivity, and we want that to happen during adaptive or physiological processes by improving muscle quality and lowering the possibility for loss in strength and function. So your patient populations, things you need to consider is old age or pre-frailty. We do know that sarcopenia is higher in older people, so we really don't want to lose muscle mass there. The severity of the disease, how overweight are they? How high is their glucose or A1C? Do the subjects have a risk of sarcopenia, or are they coming in already sarcopenic, and we really need to watch the monitoring of the muscle? And then, is there a way to improve muscle mass while subjects are on these GLP-1s? Because the combination of weight loss with muscle mass gain or maintenance is a really important outcome for those subjects on trial. So let's go on and talk about DEXA scanning here, because DEXA scan, I think this is your best bang for the buck approach to using imaging in a trial looking at body composition. Now, I know we do get bone mineral content, and that's very important, but it's the combination, at least in the beginning phases of efficacy, that I'm looking at what is my percent change in lean and fat masses across my body. I can do this regionally. I can look at the arms or the hips or the legs. I can look at the bone composition over time with a really long timeline. Again, I'm talking about 1 year here. Or I can look at the visceral fat analysis, which has been validated in DEXA against CT distribution. So that's a good endpoint as well, since we know about inflammatory mediators in this space. DEXA is widely available. There's a high patient acceptance. There's no claustrophobia. We can scan really big people by doing just half the body. Luckily, there's a high amount of bilateral symmetry, so a really big person can be scanned. It's accurate and precise. It's highly reproducible, but it is an indirect measure of adipose and muscle. So let's go on, please. And we're going to look at muscle mass, weight, and insulin sensitivity. We want to maintain muscle mass and the bone density, but obese people tend to have poor muscle quality. So that functional aspect is really important. We know that skeletal muscle regulates insulin-mediated glucose transport through the glucose transport receptor, and we want to be driving glucose into the muscle. In a weight loss trial, even a small amount of weight loss can improve the metabolic rate, skeletal muscle quality, and enhance adipose tissue with liver insulin sensitivity. Insulin, on the other side, suppresses the muscle proteolysis, whereas insulin resistance plays a role in that sarcopenic obesity, where measurements like weight, height, and BMI are ineffective. So let's go on and bring in a new imaging modality here. Let's talk about MRI for whole body, and then specifically for the SAT-VAT measurement. So whole body, this is a solution by AMRA. We do multiple acquisitions. They can segment out the adipose and the muscle and then look at the fat. They can do this by volume. They can do it by regions, just like I can with DEXA, looking at the arms and the legs and the trunk. I'm going to talk about ectopic fat later on, and then just looking at the SAT-VAT ratio. This is the sub-Q versus visceral fat. That's the bottom of the image here, where the yellow is the VAT and the purple is the SAT. And we find in acquisitions that it's really just a single slice is all you need, but you're going to go ahead and acquire multiple slices if you wanted to go back and do a volume analysis, and you can do a whole abdomen acquisition as well. We find that using a spinal landmark is better for repositioning, because if a patient loses a lot of weight, their umbilicus is in a new location at follow-up. We find that their spine actually doesn't change too much. So these are direct measures of fat. And of course, we want them to be accurate and precise between those visits. Now, the downside to doing an MRI analysis is it's expensive. There's a complicated acquisition protocol, and that we do find really big people have a hard time fitting into the magnet, or for us to get a whole a field of view. So you may be excluding subjects that are too large for your trial. So let's go on. Thank you. So let's talk about visceral fat here. You know, we can use waist circumference as an indirect measure of visceral fat. This is easy and it's inexpensive to measure, but it is not considered a surrogate for visceral fat content or metabolic abnormalities, because the procedure itself is not standardized. So you really need to look at, is this a measurement that's impacted by the extent of non-visceral fat? And then what's the accuracy of looking at visceral fat when you have patients who have a very high BMI? So let's go on and talk about some of the other areas that we can do. Ectopic fat. For a long time, hepatic fat fraction, using the proton density fat fraction technique, it's a 3D method, limited to sites with the advanced liver packages. That was, that's one of the ways that we can look at fat. I can also look at intramuscular fat using Dixon T1 weighted images, looking at sub-Q, intra and intramuscular fat. I can also look at fat around the pancreas, the kidney, the heart, epidural fat. These are widely available sequences, other than the PDFF, which requires special scanner software. They're accurate and precise measurements. We tend to exclude large subjects, and I can do this with CT, but I really only look at muscle attenuation. So let's go ahead. Thanks. And just wanted to specially call out cardiac health here as an endpoint, because this is one of those, I'm going to use GLP-1, but I'm really looking at heart health. And this has to do with the changes related to epicardial fat and pericardial fat. And if I can change those pools over time, that I can alter the functionality and the mass of the left ventricle and the whole fat in the chest, that's called pericardial, that gets reduced as well. So these changes may contribute to the reduction in heart failure events. Downside to this is cardiac MR. While accurate and precise, it's a manual segmentation technique and it's very expensive. Cardiac MR is always more expensive than echo. So you got to weigh that as well. Let's go ahead. I'm getting close to my end here. Okay, this is another area when I can look at, when does imaging tell me about activity? Here was a study where we looked at blood flow in the brain. This is functional MRI. Patients were shown images of a high-fat food like French fries, a sweet food like chocolate cake, steak, and something neutral like a red stapler. They were given a rest period and then challenged again with those same images and then rested. And this challenge versus rest, we could demonstrate that over these fMRI images in various regions of the brain, tirzepatide treatment reduced brain activation for food, but it didn't do that in the placebo group. And it also didn't do it in liraglutide, which is also in the GLP-1. So there are differences in brain activation, and that may be important for you to help you differentiate your drug versus some other technique that's out there. So I've got, I think, one more concluding slide. Thanks. So again, if I think about what are the lifestyle modifications that need to occur here, yes, we can image muscle and fat, but you also need to make sure that you're preserving that lean mass and reducing the negative side effects. How you do that is with diet, exercise, maintaining strength, and making sure that you're monitoring your sarcopenic obese patients so that they're not going into a crisis, if you will, by losing too much lean mass. I can also, by changing my diet around, change my secretion of insulin and also encourage endogenous GLP-1 release endogenously. We may see in the future, in addition to resistance training, the addition to GLP-1s of other molecules that are designed to promote muscle growth or stability. And I think my last kind of statement here before I turn things over to my friend and colleague Judy Nerisi to say that the maintenance of muscle health is critical during treatments to improve glucose management and or weight loss. So take it away, Judy.
Thanks, Mark. So now I'm going to talk a little bit about adjudication and GLP-1s, but in order to do that, I think we really need to talk about and understand what adjudication in clinical research is. So you can move on to the next slide. And so clinical adjudication is a review process by clinical experts in that therapeutic area of predefined events in an independent and unbiased manner. It applies a consistent definition to each of those events, and it actually minimizes bias and provides integrity and reliability in the data. So it helps support data-driven insights and regulatory approval. We can go to the next slide. And why do we adjudicate? So we adjudicate because it helps reduce variability across sites and regions. It protects the data accuracy. It minimizes unintentional bias that may be there from the site reporting. It helps to decrease inconsistencies in your data. And regulatory authorities seem to be recommending adjudication of events because they recognize its role in robust and unbiased data. So so now we're going to talk about in the next slide where where the FDA guidance has come out, and it typically in GLP-1 studies, it's cardiovascular. So cardiovascular outcomes trials have been around in GLP-1 studies for quite some time, and it's historically where GLP-1 adjudication has occurred. So let's move on to the types of, so in GLP-1s, the major adverse cardiac events, this is a list of them. What I've seen is myocardial infarction, death, strokes, heart failure, hospitalization for unstable angina, and revascularization. In addition to these cardiac events, we've also looked at pancreatitis, medullary thyroid carcinoma, and some renal events. Move on, please. Adjudication does not just occur in safety, but it also occurs we do efficacy adjudication. And in one international double-blind study where we had 731 heart failure patients with an EF of at least 50% and a body mass index equal to or greater than 30, received tirzepatide 50 milligrams weekly for 52 weeks. The two primary endpoints that were looked at during this trial were the composite of adjudicated deaths from cardiovascular causes or a worsening of heart failure for them. So the result of it showed that a total of 364 patients that were assigned to the tirzepatide group, of that number, only 36 patients had an event, or 9.9%. And in the placebo group, we had 56% or 15.3% that had an event. So the conclusion from that study was that tirzepatide led to lowering the risk of composite death from cardiovascular or worsening of heart failure. And the adjudication of those events helped lead to that conclusion. So it can be quite useful. Next slide, please. So how does GLP-1 trials work? So there's been numerous studies in GLP-1 studies where for safety and accuracy. Key to these trials was the weight loss, cardiovascular protective. It is in obese patients. And they're still not fully understood how that cardioprotective protectiveness occurs. So is it just the loss of the weight and the body fat? Is it a renewed attitude towards their health and their psychological? Is there a psychological benefit to it? Or is there some kind of mechanism or impact on the myocardium that we have yet to investigate? So in the future, I can see perhaps those types of endpoints being looked at. Next slide, please. And I would be remiss if I did not talk about medullary thyroid carcinoma in a GLP-1 adjudication talk. So there's really no definitive data that shows that GLP-1 agonist treatment correlates to a medullary thyroid carcinoma. There is caution highly recommended in subjects who have a family history of medullary thyroid cancer, but there has to be more methodologically superior studies long-term of these patients to really clarify the risk versus the benefit of GLP-1 agonists in these types of patients. Next slide, please. So key takeaways of clinical adjudication. It provides greater accuracy, it's standardized event classifications, it's objective and consistent because it's an independent process which provides unbiased and independent review of these events. It aligns with standards and meets regulatory expectations. And just remember that expertise matters, that you want to have skilled adjudication team that ensures the unbiased and rigorous assessment of these events. Next slide, please, Ryan. I think I have a summary. Good Lord, that summary is tiny. So in summary, there's various ways to access body compositions in GLP-1 study trials. And the FDA guidance suggests that DEXA is the modality. MRI methods can provide outstanding supportive data. Clinical outcome assessments play a crucial role in developing a robust GLP-1 endpoint strategy, and a comprehensive safety strategy in GLP-1 clinical trials should include electronic patient-reported outcomes. And then a robust adjudication of events of special interest utilizing a 21 CFR compliant electronic platform is really helpful, and having a knowledgeable adjudication team is paramount. So I'm going to punt it back to you, Ryan, for question and answers.
Well, thank you very much for that very insightful presentation from all of you. I have at this point like to invite the audience to keep sending questions or comments for this Q&A portion of the webinar. If you're attending via GoToWebinar, you can use the questions window. And for those watching on LinkedIn Live events, feel free to post your questions in the comments section. Now, I've already got some questions from the audience, though, so we'll get ourselves started with those. The very first question I have here, Jessica, I think this is for yourself. Someone just wants to know, how does the ECOA strategy change, or what different considerations are needed when the target population is pediatric?
Okay, that's a good question. So I guess the considerations are broadly the same in my mind, but some of them really come into the spotlight and are much more complex when you're talking about pediatric participants. So for assessment selection, you'll want to make sure that you're selecting surveys that are validated for the right age group, because not all surveys are validated for all ages. And you'll probably have some aspect of a caregiver contributing to that data. Maybe they'll complete assessments on behalf of the pediatric participant. So not all surveys are validated for that purpose either. And you can imagine that the at-home data collection piece comes into play when it comes to complexities as well. in a pediatric population, right? So regulatory authorities really expect that data to be accurately attributed to whoever is providing that data, and so the system needs to account and track for that. And then the last piece I'll say on that is really the training component. So I can't stress enough how important it is when you basically send people home with homework to do that they need to comply with. It becomes really important for them to have at-home instructions. And so those instructions may need to be directed at the caregivers, but if the pediatric participant maybe is self-reporting some data, it also needs to be content tailored for them as well, and keeping their level of literacy in mind. So those are some areas I think would be key for that.
All right, thank you very much for that answer. I've got a question here, wondering how do we choose between the various imaging modalities that can be used in a weight loss trial?
Yeah, that's a great question, because there are so many options. When I look at DEXA, MR, CT, do I do regional analysis? Do I do whole body analysis? It really comes down to what stage are you at in your development? What's the development question at hand? And maybe more importantly, what's the timeline of exposure? And we've got to put it on the table, is what's your budget? There's going to be strengths and weaknesses of every imaging endpoint and every imaging modality. And that goes to the sensitivity to change, as well as, even if you want to do it, can you set up enough sites to be able to do it? And can they submit the images in for central review? So there's a lot of considerations, part just feasibility, and then partly related to budget. If you are thinking about a phase 2 strategy that gets you ready for that end of phase 2 meeting that takes you into doing DEXA in a long trial, for example, and say, hey, I want to do a subset of my subjects in MRI, that's where you bring forward all the investments that you've made in, say, an MRI technique that still does DEXA plus MRI, maybe a subset. The guidance doesn't really say what that subset looks like. They have a number for the efficacy part, but the imaging is just really a subset. So that's part of your sensitivity to change with your dosing regimen, PK/PD. That's what you need to be factoring in your, again, overall strategy, not just a linear approach to study by study.
Perfect. Thank you so much for that answer. Now, Judy, I've got a question here for you. Someone's saying that when thinking about implementing an adjudication process for endpoints in a study, what are the typical rate limiting factors that you see that could impact the turnaround time or slow the adjudication process?
So I'm going to tell you the number one rate limiting factor that we see is the site's ability to provide the source documents for the event itself. So if you think about how adjudication occurs, it's not a timed event. It's an event that occurs at any time during the study. And once that event occurs, you need to be able to supply the source documents from the treating facility to the adjudication team for review. So that is typically the rate limiting factor. So if you think about site training and site selection, they are really important at the onset of your study so that your sites know the expectation when an event occurs and have the ability to upload and supply those source documents to us.
Wonderful. Thank you so much for that answer. Now, Jessica, I've got another question coming in for you. Someone wants to know just what are the ECOAs that you'd recommend in a protocol to robustly evaluate and then therefore differentiate based on depression or anxiety, for example?
That's a good question. There are a lot of assessments around depression and anxiety out there. Some have better construct validity and reliability than others. So on the clinician reported outcome side, there are the Hamilton scales, the HAM-D and HAM-A as they're called. And then you have some participant reported scales. The HADS is one, the hospital anxiety and depression scale. That's used across a lot of different indications and seems to successfully detect change pretty well. Yeah, while we're on the topic of mental health, I think, you know, it's important that this is thought about not only for efficacy, but also safety data. So you can think about, you know, including suicidal ideation and behavior assessments as part of these trial protocols. Of course, you know, there's only so much time, and I didn't get to touch on this too much yet, but I can mention it here, right, that there are scales like the Columbia Suicide Severity Rating Scale, or the CSSRS, or the PHQ-9. Those are just a couple validated options that are really expected in safety data for these trials as well. Yeah.
Okay, thank you very much. Now, Mark, a question for you here. Someone saying, considering the different endpoints, what guidance could you offer as to the optimum time requirements between visits in a GLP-1 trial?
Yeah, timing and sensitivity to change are really important. If I'm in say an early phase 2, I may only have safety coverage that allows, say, 24 weeks of dosing. Now, in that case, I'm going to look for what's the modality that's going to be the most sensitive to change. I would probably choose MRI. I may do baseline week 12, week 24. If I do DEXA in 24 weeks, I can get body composition, but I'm probably not going to see much on bone mineral density. That's where studies that go out to 52 weeks, I could have a combination of MRI and CT at, you know, maybe every 3 to 6 months as intervals. I want to have decent coverage, so I'm sensitivity to change, so I can understand my rate of change. At the same time, not overburden subjects with having them coming in for imaging on a so frequent basis to the point that they drop out because they no longer want to be in the magnet, or they don't want to have a DEXA scan done. So that's always a consideration for anybody on a trial, is when you make your schedule of activity, what are the procedures, and then what are the time points that you want to use? And if you're not sure what would be some possible you know, timing of those imaging events, just let us know, and we can work with you on what would be a decent imaging design.
Great, thank you so much. I've got another question here, wondering when setting up an independent adjudication process for a study, Judy, what are some common challenges that you see during the setup process that can impact timelines?
So one of the biggest considerations I have is sponsors typically don't have a full understanding of what the adjudication process is and what the standards of adjudication are. So working with that sponsor and helping to guide them to understanding the standards that we need to work under, as far as the independence of the adjudication committee, as well as providing that committee with the unbiased data for them to make their assessment, is really important. And then while we're doing that, we're also working on the charter to make sure that, you know, we define everything clearly, so that once that process begins, the adjudicators have a clear definition of what they're adjudicating, because it can be a little different. A definition for an event can be different from one study to another, and what they're looking at and trying to analyze can be a little different from one study to another. So that's really important. And then again, I can say one of the biggest challenges is making sure that your sites are the correct sites that are able to clearly upload, you know, easily upload and not have such a burden on the site. So sometimes, like some of these some of the ePro data and the imaging data is really helpful to us. We're able to, you know, utilize some of that data as part of our adjudication supporting documentation. So it's very helpful.
Wonderful. Thank you very much for that answer and for all of the answers today, but we have reached the end of our time here today. For those who are interested in hearing more from Clario, you can register for their next webinar, The Evolving Global AI Regulatory Landscape, Recent Developments and Practical Insights for Clinical Trials, which is scheduled for Thursday, March 20th, 2025, and you can use the link I've sent in the chat box or the QR code on the screen. Also, if we couldn't attend to your questions, know that the team at Clario will follow up with you, or if you have further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've also shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event to gain access to features such as viewing the recording and more. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you.